So Sam, I'm going to go ahead and introduce Jay. So Jay Russak is currently Director of Clinical Development Operations at Dicerna Pharmaceuticals, where he is focused primarily on clinical development strategy, trial design, and execution of their lead program. Jay has 14 years of experience in the field of clinical research, 10 of which have been spent directly managing clinical trials in small and technology companies. He has experience planning for and being part of the FDA, MHRA, and EMA inspections. Before his current role at Dicerna, Jay held roles of increasing responsibility at Finch Therapeutics, Sage Therapeutics, Biogen, and the Dana-Farber Cancer Institute. Jay has a BS in biology from Northeastern, and a master's from Boston University School of Medicine, and an MBA from WPI. So Jay, looking forward to your talk.
Thank you so much, Dr. Tata. So I will go ahead and start this presentation. So just can I confirm that everyone can see the slides? Okay. You see the full presentation mode, not the speaker mode, I hope?
Yep, full presentation.
Awesome. So I'll go ahead and get started. So today I'll be discussing using data effectively in studies with smaller numbers of patients. Quick disclaimer that the views presented today are those of myself and not necessarily those of Dicerna, my employer. I will skip through this. Dr. Tata did provide my background. So you could see I've been in the industry for 14 going on 15 years and have worked on a varied number of organizations. Most recently, the past few years have been in smaller companies where we focused on some more smaller indications, which led to the genesis of this talk. So I have three main objectives for this brief talk this morning or this afternoon. I'd like to talk about how ensuring data is accurate and efficient in situations where we only have one attempt at a trial as possible. So when you're working for smaller companies or on a rare disease, oftentimes you don't have the opportunity to, get it wrong. Even in larger companies, once you get it wrong once, you might not get the approval, resources, and money to go ahead and do it again. To optimize ways of analyzing data to ensure regulatory guidelines are met, even with those smaller sample numbers and single-arm studies. And then strategies and solutions to maximize the data obtained from orphan drug trials. And I have several examples of different adaptive trial and covariate adjusted randomization designs of some examples of ways to not only move a little bit faster within the development timeline, but also with a lot smaller sample sizes. So whether you're a small biotech or a large pharma, rare disease trials with limited patients often only have one opportunity to get them done right. You literally have just that one shot on goal. Especially with the increasing cost of trials, companies cannot afford to have to repeat these trials. In addition, with an increase in the competitive landscape, even in an orphan disease indication, it adds to the pressure of getting the trials done right and in a timely manner the first time around. A theme you'll hear me discuss throughout the next few slides is the need to plan and be smart about your plan. Use all the resources at your disposal. Go and find even more to ensure you are leaving no stone unturned, and even go down the path of developing a new treatment for a rare disease. The importance of identifying go, no-go criteria and predefining them and sticking to them is also critical. I've often seen the pitfall of a development organization is when you think you have them defined, but then you keep on moving those goalposts as you get new, maybe unexpected or undesirable data points, and then you're updating criteria and endpoints along the way. Yes, there's always exceptions to the rule where this is acceptable, but being diligent to predefine the right critical success factors, and if you happen to fall short, be confident in saying it is a no-go decision. So, discussing your plans with regulators. While common indications can receive special designations, as the feature of my talk is really focusing on the smaller sample size studies often found in rare disease, it's a lot more common for rare disease programs to be granted with a number of different special designations or statuses. whether we're talking about breakthrough therapy designation, orphan status, prime status, etc. These designations and statuses are in place to help sponsors increase the likelihood of success of getting from the point of a marketing approval submission, and in some cases doing it significantly faster than the traditional path. Be strategic with how you utilize these interactions with the regulators. Take the opportunity to meet with them as often as you can. And when new questions arise, don't leave you and your organization guessing on what you think the path to filing is. Ensure that they agree to the plan and how to get from phase 1 to a pivotal study, what your critical endpoints are, and how they will be measured, and any other key details related to your development program. Excuse me. Given the small data sets and the one chance we have at it, it is important to ensure the quality of the data as good as it can be. Here are a few operational aspects that I commonly deploy across my programs. So we'll start first and foremost with setting the expectation with your CRO and any other key vendors, such as the labs related to your primary endpoint. The data quality is of the utmost importance, and one avenue or one process to put in place to help ensure that it stays high is introducing quality agreements with these key vendors. A quick 2 words of advice based on my experience in terms of setting up these quality agreements. You want to make sure that they're simple enough for all to understand and easily measure against. I've often seen too many times where quality agreements become overburdened with long lists of KPIs and metrics that are going to be measured to track the health and status of the relationship or project that you're governing. This becomes overwhelming and difficult to come away with a clear reading on really what is the state of your project. And ultimately, many times you fall into what we all like to say, paralysis by analysis. So I would urge you to keep these quality agreements simple. Identify the top critical aspects of your study and what the key success factors are, and pick only a few metrics to measure. Moving on, while we could spend hours debating the pros and cons that technology and clinical trials offer, the use of technology to improve the data quality is worth spending a moment on. I personally hope that one of the positive outcomes from this pandemic and our heavy reliance on technology, remote source data verification, and digital data is innovations that reduce activity, such as the transcription of data that is already recorded elsewhere, like the EMR, electronic medical record, into our CRFs. Think about how much time our site staff and CRAs spend transcribing and then source data verifying the data when it already exists elsewhere as the authoritative source in the hospital or clinic's electronic medical record. Yet here we are spending hundreds of thousands, if not millions of dollars, accepting the current standard practice of being for all data to be manually entered in by the site into our databases and then source verified by the monitors that we either employ or hire via the CRO. There needs to be a better way to go about this. Another aspect of technology which has been slow to be adapted is the use of wearables. Yes, many of us on the call today have introduced the use of wearables into our studies in one way or another, but there are often many gaps and limitations at the end of the study when you're trying to analyze that data. And it becomes quite complicated. And I know I personally have questioned at the end of a study, did we really add any value by adding these cool and fun yet expensive devices? I think we have a long way to go as an industry before we see wearables truly deliver an efficient method of real-time data collection of that key important data. Next, one of my favorite processes is rolling data cleaning. Not only will that give you an ongoing insight and ensure quality is being met along the way, but it will shorten your time at the end of the study from last patient out to database lock, as the goal is to have data lock ready along the way as patients move throughout your studies. So in essence, as patients are completing those time points or those milestones, you're reviewing it, querying it, and soft freezing it or soft locking it, whatever term you want to use or your data management group wants to use. So at the end of the study, you've basically seen all of the data except the last visit or last couple of visits, and you're doing one final review and able to quickly lock that database. Last but not least, let's talk about building strong site relationships. There are many pros to dedicating the time to build these relationships, but with relation to data quality, it's pretty simple. I started off as a study coordinator at Dana-Farber, as I was introduced, and I could speak from that experience that it's often true that study coordinators who like working with you because they value their relationship with you are going to try harder and do a better job at their overall responsibilities, the stronger the relationship is. I know this seems like common sense, and it is, but in my experience, often we try to unnecessarily complicate common sense. It will also help when you need to get a site to respond to queries, for example, or clarify something that you already have that strong relationship. So moving on into the statistical considerations, and then in a few slides, those examples of study designs and how we could use, how could we effectively use smaller amounts of data to still be effective in our ability to have a data set and a set of studies that are deemed ready for submission for marketing approval. So trials fail often because the wrong question or too many questions were asked, or in other words, the endpoint or endpoints chosen aren't able to show the hypothesized effect. Yes, not all drugs work, and as an industry, we have an inherently high failure rate. And there are papers that outline this, that often it is not the actual drug, but it's actually the design of the study that failed. And a different approach would have led to a lot different outcome, i.e. a successful readout instead of a failed trial. So if we look at the top of the slide, the enrollment rate directly dictates the study duration and is driven by the sample size, which is calculated dependent on the necessary powering of the study. These are all quite interrelated. Basic 101. But hit the wrong button there. But as we work our way down into the middle of this slide, we talk about a few aspects of how statistical integrity of a study can be attributed to, including how representative the study population is to the target and the accuracy of our inferences when designing the study. For example, how representative is the study population when we look at the eligibility criteria? What are those IE criteria that we're setting? Is it really representative of that target population, or have we fallen into the trap of what's been done before, that all phase 1 studies have to have, or all phase 2 studies, or whatever phase, have to have these basic sets of IE criteria? Are we using the right inferences? Are we looking at the right historical sets of data to define to justify what the design of the study is and how we justified the sample size and the power calculations? So some of these key attributes and the relationships between them are shown at the bottom of the slide. So for example, one way to increase the statistical integrity is to increase the sample size. More patients, you could say that the outcome is more reliable. How often? The topic of this talk is how to use smaller sample sizes. And in rare disease trials, you often can't find the initial set of patients you need, let alone even more. So other things that other attributes that we need to look at and consider when trying to keep the statistical integrity of the study high, however, decrease the overall sample size. So one way is about the informativeness of the endpoint. And this is that theme that I've talked about, is being smart and picking asking the right question from the beginning. What is that critical endpoint that needs to be answered to prove that your hypothesis is right with your drug? Obviously, we also want to find ways to reduce the placebo rate. While placebo-controlled trials remain prevalent in our industry, we know that there is that ongoing ethical debate that often occurs is it ethical to expose patients, especially in a rare disease setting, that have no other treatment options, to a course of placebo to be able to effectively show comparisons between your drug product and the control. If you find ways to lower the placebo rate, such as looking at adaptive randomization and with real-time monitoring, which I'll talk a little bit more in a couple slides, there's ways to actually decrease the placebo rate and transition more of your patients enrolling in the study, if the study is showing to be efficacious, to the active arm, and thus hopefully will increase enrollment, as if it is a competitive landscape, patients will be more interested in a study where they have a higher likelihood of being assigned to active than placebo. So as we look at sample size and enrollment rate considerations, the overall goal here is to lower the sample size as much as possible. So I'll show some examples of study designs that aim at reducing the sample size by utilizing historic data and thus reducing the number needed in the placebo arm, as well as utilizing adaptive design to shift the randomization during a study. I won't speak to the enrollment rate, but do want to spend a moment on the optimal study design and factors contributing to the sample size. This strategy speaks to being smart and efficient, ultimately being able to identify not only a big winner early, but equally, if not more important, a properly identifying a loser as to not expose more patients unnecessarily to a drug not working and utilizing more resources than needed on a drug that doesn't work. Items like treatment effect, endpoints, and adaptive design can all aim at lowering the overall number of patients you need, but still have a powered data set worthy of submission for approval. So as we look at this next slide, I quickly want to highlight the difference between seamless and accelerated design when compared to traditional designs. And then my following two slides will talk about two examples, once again, using Bayesian statistics and covariate adjusted randomization techniques. So traditional design would be to complete your phase 1 studies, your SAD, MAD studies, additional phase 1 studies to narrow down the dose or doses you want to bring into phase 2. You have your phase 2 trials, which act as your proof of concept that your drug has its desired effect, and hopefully no new safety concerns are popping up. You then move into your set of pivotal studies. As you move down the stages of these studies, from phase 1 to phase 3, your sample size requirements often increase. This all becomes challenging in a setting of a rare or orphan disease. When we look at accelerated and seamless designs, those are two examples of ways to shorten the overall development of a drug and try to reduce the white space that occurs in traditional development. So if we start on the left-hand side and look at accelerated, so you still have two separate studies, but it reduces that said white space in between the decision to start phase 2 because you're using a limited data set. You're not waiting for that complete data set to read out. You're looking at an interim analysis in the phase 1B aspect of your program, and then making that determination of whether or not you have a go decision to initiate your phase 2 as your phase 1s are wrapping up. There's lower regulatory risk. This approach has been shown, and there's plenty of study designs or development pathways that have utilized this accelerated approach. You often pre-negotiate this with your agencies when you're meeting with them on what is the agreed upon path and what data do you need to show to go from, the first study to your second study to your third study. The cons of accelerated, though, as shown on the slide, is, it does carry uncertainty to the design features, uncertainty as you get up to your IA interim analysis time point, as well as you're still making a decision on a limited data set. You're not as informed as your traditional design. If we switch over to the seamless on the right-hand side of this slide, you're combining basically two studies into one singular protocol with predefined rules of when to transition from the earlier phase to the later phase. The obvious pro is that this is the most efficient method of reducing the white space. However, there are some additional cons which come inherently with this approach. You're moving along with limited data, and you're also increasing the complexity of your regulatory interactions as you operate under one protocol. So if there's feedback on a specific earlier phase or later phase of your design, you're still amending this one overall document. And from an operational standpoint, for any of you of those in ops or regulatory, we know that this is adding potentially unnecessarily extra work for the team. So here's an example where we compare a phase 2 design that uses both Bayesian statistics and dynamic borrowing along with covariate adjusted response adaptive plans. So we were looking at how do we take in a rare disease setting, how do we transition from a phase 1 to a phase 2 where we could quote unquote catch up or stay competitive with others in the space, and reduce the overall number of patients that we need to enroll. Traditional design of the trial, we were looking just for numbers. For example, we were looking, let's say, at needing 100 in the treatment arm and 100 in the placebo arm, so a total of 200 patients. And as we went through this applying a Bayesian with dynamic borrowing approach, we were able to reduce and determine that we might be able to, or we would be able to enroll anywhere between 90 to 120, 150 patients. So we're reducing it we aimed at least by 50% of the sample size as a rough estimate by deploying this design. What dynamic borrowing allows us to do is the use of historical data that may be present from registries and other data sets to better inform the assumptions and thus lower the overall sample size needed of both the treatment and placebo arms. The adaptive randomization aspect of the design utilizes real-time monitoring, as I've discussed earlier, of the data to inform and update the randomization schema and reduce the placebo arm if the treatment is having the desired effect. So you would have a separate unblinded team that would be looking at predefined time points and predefined data sets that show, is the drug having its desired effect? And if it was, there would be an update that's already predefined in the protocol that would allow you to basically reallocate your stratification, and instead of maybe 2 to 1, you could then make it 3 to 1 as an example. So you're allowing more patients, because you're already seeing an early desired effect, to be assigned to that treatment arm instead of the placebo arm. The slide goes into specific details about, what are the risks associated with this design, which include complex logistics, as well as needing for that continuous monitoring with an independent kind of monitoring board, as well you also need the right historical data to perform the dynamic borrowing. So that's one of the big differences between this slide and the next slide. So once again, dynamic borrowing, you're using a set of historical data to make assumptions and to inform your sample size analysis. If you happen to not have that set, so maybe you're working in a rare disease that you're the first there, and there is very limited to no historical data that currently exists in literature that you could apply dynamic borrowing for. So this is a CARA design, covariate adjusted responsive adaptive design, where we're still able to lower. So instead of 200 total subjects, we estimate between 120 and 150 patients would be needed. Every patient matters, especially when we're talking about orphan or rare disease. So the fact that we're still able to see this reduction in patients, because once again, we would be taking looks at real-time looks at the data and being able to have real-time indications, is our drug having its desired effect? As also highlighted previously, not only does this help identify an early winner and help expose more patients to a treatment that is showing success, but also on the converse side, if you happen to be unlucky and the drug's not having the desired effect, you could more quickly shut down the trial than if that is indicated. So I've talked about real-time monitoring. I want to just talk about kind of some of the key aspects you should be thinking about of how to deploy real-time monitoring to enable that adaptive design. So the left-hand side of the slide is the standard monitoring that occurs on most studies. You have your risk-based monitoring, source data verification, safety monitoring, KPIs on site performance, et cetera. But if we focus on the right-hand side, as I said, it goes into predefining what data we want to have a look at, such as key biomarkers or clinical assessments that can help provide the answers on, can we shift the randomization? Should we perform an interim analysis? Do we have enough data that will actually show a meaningful analysis? Or are we ready to move on to the next phase of the study, et cetera? So this all is the basis of that covariate adjusted randomization and attributes to be considering when setting up this design. So in summary, the challenge in conducting clinical trials in rare diseases is the small sample sizes and thus limited sets of data that we have to work with. As I discussed, utilizing increased access to align on strategy with regulators continually along the way in addition to utilizing newer study designs such as adaptive randomization. And as always, continuing to find innovative ways to ensure the best data quality you can will result in having a complete submission package for the hopeful approval of your product. And with that, I would like to thank you for your attention and welcome any questions. I also welcome you to contact me after today to continue this conversation. So thank you.
Great. Well, thank you very much. I think we do have one question from Adriana Camprelli. And that is, can you speak to limitations imposed by FDA for sample size? This is rate limiting and expedited drug development with a non-specific IND.
Sure. So we specifically, over the last couple of roles, have had quite a bit of, we've had the fortune of having some of those statuses where we get to interact with the agency quite a bit on our thoughts. I could say that specifically with the limitations imposed regard to sample size, it's getting to the table and discussing, what is your approach? What is your thoughts on these maybe newer design attributes, and understanding what they're thinking of. What are they? What's their lens that they're looking through versus what yours? I would definitely encourage you to ask those questions on why are they imposing limitations that you are hearing from them. Try to understand the why behind their rationale, right? I think we've had a lot of success on carefully but strongly challenging some of their thoughts on ensuring that, but you have to go to the table ready with and backed with your data to support why you are challenging them. Challenging sounds like a hard word, and you don't often want to challenge the FDA, but why you want to propose an alternative method or alternative approach to what they are maybe recommending. It's also that balance, right? Give and take. I've often found with the FDA and negotiating the path forward on your clinical development plan. So what's your path to approval? Identify as an organization what you're willing to negotiate or kind of concede to, or maybe, meet in the middle versus what are things that you feel are make or break for your program, and what are you willing to, kind of stick strongly to. Those have been tactics that often have worked well with our personal interactions that I've had with the agency on my programs.
Mm-hmm. Okay. And I think, as you talk about these adaptive design trials, one of the ones that I really think are good examples of sort of these multi-arm trials that have common control groups are, for example, the REMAP COVID trial and GBM Agile, iSpy 2. I think that those are all really, the types of trials that, require a lot of collaboration among companies, but I think that are really great for patients.
Absolutely. I think we're going to be forced to have more collaboration in the industry, more collaboration amongst our peers, as well as I know you always have to put our patients first. So I think this is all in the effort. Yes, we're looking to bring more drugs to market ultimately, so these patients in need have more therapies and access to them, especially those that are orphan indications and don't have any other options. So absolutely. Yep. I see one other question popped up.
Mm-hmm. Yep. Go ahead.
So the question is, can you speak to more successfully enrolling food effects sub-studies in phase 1B studies? Sure. To be perfectly transparent, I don't have a ton of experience with food effects sub-studies in phase 1B. I haven't had the need, fortunately, on the current and past few programs I've worked on to do a lot of food effects sub-studies. So I'm going to default. I don't know if Dr. Tada, you have any insight, but unfortunately, I don't really have a good answer for that. I apologize.
No, I think some of it could be, if you're able to go into a healthy volunteer population, that's always better. But if you have to go into a patient population, those are, those once you have your recommended phase 2 dose, you may want to go, start enrolling at that point. Yep.
Sure.
So again, well, thank you very much. We appreciate your time, and I think Sam, you're going to transfer us to the next room.
Yeah, I appreciate your time again, Jay. It was a very interesting presentation again. And yeah, I will be. There is one more comment by Monica Pudney, VP ClinOps from Metabomed. She said, "You amend the protocol by adding a parallel arm." So maybe been a comment. But yeah, with
Oh,
that,
that may have been a comment to the
Food
phase,
effect.
the food effect question.
awesome. So again, thank you, Jay, and I will be transferring everyone to the next session.
Thank you so much.